Assalamu alaikum. In this lecture, we will discuss some quiz related to phospholipids and carbohydrates. So let's start. Which statements about the differences between the phospholipids and the triglycerides are correct? Phospholipids have hydrophobic regions, but triglycerides do not. But triglycerides also have hydrophobic regions. So this statement is incorrect. The fatty acids in a phospholipid are always saturated, but in the triglyceride they may be saturated and unsaturated. But in the case of phospholipid, they can also be saturated and unsaturated both. So phospholipids are the polar molecules, but triglycerides are non-polar. Yes, this is the true difference. So option D here is the right answer. An investigation was carried out into the effect of the different treatments on the permeability of the cell surface membranes and tonoplast of beetroot cells. The beetroot cells vacuoles contain red pigment which which cannot be diffused from the tonoplast and cell surface membrane. So a particular cell of the beetroot tissue was cut and then it was placed for the 20 minutes to remove any in the water to remove any of the pigment, right? So then the cubes were placed in the different solutions and the observations were recorded. So let's check what treatment gives the correct result and observation and explanations. Dilute hydrochloric acid. When the beetroot cells were treated with dilute hydrochloric acid, then the contents of test tubes stay clear and the membrane proteins have been denatured. They do not denature because we are using just a dilute hydrochloric acid and it is not as much active as the concentrated one. So all the proteins will not be denatured here. So option A here is not correct. Let's discuss option number B. Here ethanol is taken, and whenever we treated the beetroot cells with ethanol, then contents of test tube turns red. Yes, they turns red because it enhances the release of the red pigment from the beetroot cells, and the lipids including the membrane phospholipids have been dissolved. Yes, ethanol tends to dissolve the lipids and the permeability of the membrane was increased and the red pigment of the beetroot cells were moved out of the cells. So the content of test tubes becomes red. So option B here is the right one. The diagram shows 3 hexose sugars, 1, 2 and 3. So which correctly shows the examples of the carbohydrates in which these 3 hexose sugars are present. As we can see here, the 1 is the alpha glucose and the 2 is the beta glucose and the 3 one is the another hexose sugar. So 1 is found to be present in the case of sucrose. The 3 is found to be present in the sucrose, right? It is a monosaccharide, it is a monomer of sucrose, and the 2 is the monomer of cellulose because it is alpha glucose, and starch, the 1 is the monomer of starch. So option D here is the right one. What can occur during the condensation of the carbohydrates? Means that during the formation of carbohydrates, large carbohydrates, it can be polysaccharides. You can say polysaccharides, right? So condensation means the joining of the monomers to form the polysaccharides. A disaccharide is produced from the monosaccharide. Yes. A glycosidic bonds are broken. No, it cannot be broken. It is not a hydrolysis process. It is the formation process. Condensation is basically the formation of polysaccharides or monosaccharides or disaccharides from the monosaccharides. Molecules of water are used. There is no need of water. Monosaccharides cannot be produced. So option A here is the right one because condensation is basically a formation process. What is true about the triglycerides? Triglycerides are hydrophobic in nature. Yes, they are, and insoluble in alcohol. Yes, they are insoluble. So option B here is the right one. In an investigation, the same concentration of the enzyme phosphorylate was added to the different concentrations of glucose phosphate and incubated at 30 degrees centigrade. At 1 minute intervals, one drop of the reaction mixture was removed and then added to a drop of iodine solution on white tile. The diagram shows the results of this investigation. Here we can see a diagram here. On the horizontal surface, we can see the concentration of the glucose phosphate per milligram per decimeter cube, and on the vertical side, we can say the time per minutes, right? And the color of iodine solution is the gray one, and that of the other one is either black. What explains the trend in the results of this investigation? Phosphorylase catalyzes a reaction between the glucose phosphate to starch. No, it cannot produce the starch in this reaction. The maximum rate of the reaction is reached at 20 milligram per decimeter cube of glucose phosphate. But this is not the maximum rate. We can find out here. The maximum rate is here. So this is not a correct answer. The substrate concentration is limiting at concentration of glucose phosphate 25 milligram per decimeter cube or less. Yes, it can be. The substrate, as we can see here, at the substrate concentration 25 milligram per decimeter cube, substrate concentration can be less or limited here, because we can see that the color is black. It means that the sugars are not being degraded by the enzyme, because the concentration, because all the glucose phosphate has already been converted, and some of the enzyme has been denatured here, right? So option C here is the right answer. Which statements about the polysaccharides can be used to describe the amylose and cellulose? Contains the 1,4-glycosidic bonds, and no, they do not contain 1,6 bonds, and they are the polymer of glucose. So option B here is the right one. The diagram shows the 3 hexose sugars, and which row correctly explains the example of carbohydrates in which these hexoses are present. In the case of sucrose, the third one is present, cellulose second one is present, and the amylose, the first one, that is the glucose, is present. So option D here is the right answer. What can occur as a result of condensation reaction? Condensation reaction is basically a production of polysaccharides or polymers. We can say the polymers, right? It is basically a formation of a large molecule. So a disaccharide is formed. Yes, it can be. A glycosidic bond is broken. No, it is not hydrolysis. It is just a formation process. So how can a bond is broken? It involves in the formation of bonds. A molecule of water is produced. Yes, during the condensation reaction, the water of molecule or HCl can be produced. If it is the formation of glucose molecules, large amount of polysaccharides, then it can involve the release of water. But if it is involved the formation of some other substance, then it can release the other substances like hydrogen chloride, water, ammonia, anything else. 2 monosaccharides joined together. Yes, so we can say that option C here is the right one. The diagram shows some relationship between the features of carbohydrates. So here one circle shows alpha glucose and 1,4 bonds and branch. So which of the line correctly relates with it? Amylopectin, one goes for amylopectin, but amylopectin do not have both alpha glucose and the 1,4 bonds. Amylopectin is basically made up of beta glucose, so it is not correct. Amylose, Yes, amylose contains the both alpha glucose and the 1,4-glycosidic bonds, and glycogen contains the alpha glucose and it is branched. Yes, it is. And we have to relate the third point with the other option. And third point goes for amylopectin, because amylopectin contains both alpha glucose and it has 1,4-glycosidic bonds as well. So it is correct. 3 goes for amylopectin and 4 goes for as it has 1,4-glycosidic bonds and it is branched and it is also alpha glucose monomers. So the 4 goes for cellulose. So option B, if I find out here, is the right answer. Till the next lecture, stay tuned and stay happy, and don't forget to subscribe my channel Biology by MK. Till then, stay tuned and stay happy. Allah Hafiz.